So today is the molecular dynamics method, which is also widely used in chemoinformatics, in bioinformatics, material science, synthetic biology, structural biology, rational drug design, and related fields, computational chemistry, when you have to simulate the interaction of the molecules in a water environment, in the aqueous environment, or you can use different solvents. It is also possible to change the solvents, adding DMSO, DMF, chloroform. It's also possible to change different solvents, but for our purposes, we are simulating the different molecules in the water because we are using the protein ligand interaction. So for the synthetic biology, the protein ligand interaction is the main system. Therefore, we have to do the water simulation environment, water-based environment, like a water box. And today I will give you the overview, what is the molecular dynamics. And the molecular dynamics is the numerical method for studying many particle systems, such as molecules, clusters, microscopic system, gases, liquids, solids, to be analyzed, to analyze the physical movements. The atoms and the molecules are interacting for the fixed period of time, and then after some time you would have a representation of this interaction as a trajectory, as a system trajectory. So you usually have very small time scale for this analysis. Usually it's only for hundreds nanoseconds, from 50 to 100 and sometimes to 300 nanoseconds for the standard protein ligand interaction, and more for some bigger systems like big protein complexes, multi-protein complexes, or some big systems like proteins in the membrane, the lipid membrane. And if you want to investigate the protein ligand interaction, so this system is also submerged in the membrane, you will put it in a water box and try to simulate at least for 500 nanoseconds to 1 microsecond time scale. Molecular dynamics is used extensively in material science, chemical physics, biophysics, biochemistry. We are also comparing these results with experimental data. Sometimes we have the experimental data and we can analyze, we can create the model using the molecular dynamics. We have a lot of expertise right now. We have a cluster, and the new cluster is running to produce high trajectory, long range molecular dynamics using some sophisticated software, which we are currently installed, and everything is running on the GPU very fast. So if I would compare the speed from the previous protocols I've used a couple of years ago, let's say 5 years ago, the speed would increase at least 5-10 times. So today, for the standard protein if you would do the benchmarking, so if for the standard protein ligand interaction, you would have only a couple of days to reach the 50, 100 nanosecond time scale. So this is very, very exciting and very, very promising technique in terms of the speed, because every time you have a new GPU, it would increase the speed exponentially, at least 5-10 times, and in the future one could also try to use the small GPU units to run even bigger systems. Probably it might be possible to generate the whole the multi-complexes or the whole cell, if one would try to simulate the whole cell in the future. Until now, people try to simulate also huge systems like the viruses, and they were actually succeeded. So here, this is the overview on the molecular dynamics and kind of where we are now and what we did in the past. So what we can expect in the future. So let's just start what we have in our system, and our system is just a n-body system. So you have usually the atoms and the bonds, and by implementing the equations, you would simulate the bond vibration, the angle vibration, torsion potentials, like torsions, rotations, the rotations which are corresponded by torsion potentials. You also have electrostatics. These are the main terms, energy terms in the molecular dynamics, the electrostatics and the van der Waals interactions. And you would try to implement this for your molecule. Then the molecule starts moving, and according to the energy, and after some time, if the energy is low, then the system would be equilibrated. So that means that the system is ready to be analyzed on the interaction. As you can see here, we have also very huge systems like protein in the bilipid membrane. It's always submerged in the water box, and you also have a lot of different ions. Sometimes, if this is the ligand binding domain, you have also the ligand which is bound to this receptor. And right now we also have projects where we have the GABA receptor which is expressed in the brain as a part of the synapse, and now we are trying to design the ligands with a high affinity to this receptor in order to modulate its function in the brain, and to treat some neurological neuropathology, psychiatric and neuropathology, like a depression. So here is a kind of a scale, and if you have physics, then of course the time scale is very small. So this also corresponds to the quantum effects. If you have a chemistry, then of course the time is increasing, but still the time is also very small. And in the biology, you would have a kind of a very long time in terms of these two fields, and even even this would also be like hours and days. And the biology is the most complex, this most complex area to be simulated in comparison of these two disciplines. For this reason, we have to do the experiments for the efficient averaging and less detail. And on the other hand, if you have physics, then you can do the simulation, and the simulation would provide the extreme details and the sampling and the parameter quality. And so we are here right now in this domain, in the chemistry domain, in the molecular dynamics domain. So this is really hard to reach the biology level so far, because as I already mentioned, we are very limited in terms of time, and if we would like to simulate some biochemical processes in the cell, like here, like protein synthesis, we have ribosomes, you have also nucleic acid, and you get different interaction levels here. So this complex tRNA and the peptide chain is growing. So this is really hard. The peptide chain is growing. So this actually is very hard to simulate. Therefore, we have to reach also the sophistication of the computers to do that, and algorithms we have. Right now, the algorithms which we are using on a regular basis in the molecular dynamics, they are force field algorithms. I will talk about it in a later slide, but I would like to mention here that these algorithms are not really good in the simulation of some inorganic substances or very complex molecules like morphs, like crystals. So it's not really working in terms of the standard algorithms very well. At least at the biological level. If you have some heavy atoms here and some molecules which have also the metal atoms, usually the simulation requires very elaborate optimization and equilibration. Therefore, usually all these algorithms are working very well when you have a peptide or you have a protein like this, and you will just put it in the water and try to simulate. So it works very well. Some algorithms also capable to simulate some ligand topology like EMBA. Therefore, we also have right now, we have the EMBA for the protein ligand simulation, which is very efficient. And we have different force field for that, for this simulation. So this is just try to try to point out that we have different limitations in the molecular dynamics. In the molecular dynamics technique. So what is the molecular dynamics? The molecular dynamics is the computer simulation of physical movements of atoms and molecules in the context of n-body simulation. So you have to memorize this term as n-body simulation. Usually if you're using the classical molecular dynamics simulation, then you will apply the equation, the Newton's equation of motion for the system of interacting particle. F equals ma, and try you will try to numerically solve this equation for the number of atoms you have in the system. Usually the systems we are dealing with are extremely large in comparison to the systems used for the DFT or for molecular docking techniques, because we are adding water, so we are increasing significantly the amount of molecules in the system. And molecular dynamics method is the deterministic method, which means the state of the systems at any future can be predicted from its current state. So, of course, when deterministic, when you're implementing some classical equations, this would be the deterministic. But in our case, we are upscaling the system, so we will add in also some stochasticity to our system, because we are using a thermostat. We also have to control the temperature, and we have to control the temperature, we have to control the pressure, we have to control the other parameters, the velocities of the system. So by adding this, we are also adding some stochasticity to the system. That's why on one hand it's kind of the deterministic system, on the other it's also a stochastic, and from this stochastic system sometimes, and this rotation are random, so they're random, but of course they're determined by the parameters we included in our system. But every time you will run the simulation, you will have slightly changed value of your parameter, and this would explain why we have stochasticity in our system. The next is forces. The force on each atom is assumed to be constant during the time interval of the simulation. And some forces on the atoms are computed and combined from the current positions and velocities to generate new positions and velocities. This would be updated in a short time ahead. So from picoseconds to nanoseconds to microseconds. As I already mentioned, we're not really going through the microseconds, although the standard trajectory so far is from hundreds to 150, 100 to a couple of hundreds of nanoseconds. And what are the molecular dynamics limits? And the molecular dynamics limits are, of course, in terms of the size of the system and the number of atoms in the system. And that means that the bigger the system is, the more time you need to simulate the molecular dynamics, and more time you have to use to parameterize the system. You have some kind of different levels of the system. Here you can see that in a very short time scale, you have a quantum system with implemented in the quantum methods. So the molecular the dynamics here is very small, from femtoseconds to picoseconds, and the system also is very small. Then you try to go from picoseconds to nanoseconds, and here you would also have a relatively small system, but this system is already big enough to be simulated by using quantum algorithms. Usually these are the proteins, some complexes, some small protein complexes, protein ligand systems, host guest systems, polymer ligand systems, polymer. The polymer should be also represent a relatively small molecule, let's say tens up to 100 subunits, even less. So the polymer is not really a big polymeric chain, but rather small and very easily simulated. Usually, I'm simulating the polymers as an example, polyethylene amine, which had about 20 subunits. So it's relatively small. It's not really represent the polymer which you have in nature, but still you could approximate the dynamics of this polymer by using the small chain. The other variant of the molecular dynamics is the coarse grain, when you have the atomic representations which are approximated by the grain, by its kind of a dummy atoms, some kind of a grain structure. So you would take the area, the molecular area, like for instance the residue, or if you have a lipid membrane. So the one lipid molecule can be approximated by let's say couple of, let's say 5, 6 different grain structures, grain structures which are connected, grain modules which are connected with each other by using some kind of a bonds, like on this picture. So you get some kind of a grain structure, and sometimes we can call it a dummy atoms, and these structures are connected to each other by using also the artificial bonds. And this, as you can see, this structure is very rough in terms of the information. Therefore, we're using this term as a coarse grain. Means that the structure is not really representing the right molecular structure, but rather the kind of a topology of the structure, but in a very limited way. So by using this coarse grain, you can upscale the system and decrease the amount of atoms, and in order to add the system different the important properties, such as to understand the protein folding, because sometimes the protein folding is the time consuming process, and you might take some microseconds to millisecond time scale to reach the perfectly folded protein, like presented here. So when you get a amino acid chain, then you would perform the molecular dynamics. Then after microseconds to milliseconds, you would have a perfectly folded protein chain. Therefore, to reach these extremely long trajectories, you have to use the coarse grain models, and this might be one way to investigate the protein folding. The other possibility you can use is the protein aggregation when you have some aggregates like in Alzheimer's disease. We have you have the unfolded, impaired, mutated and impaired proteins which are aggregate and they produce some kind of a plaques, protein plaques. So this aggregation mechanism, like amyloid aggregation plaques, then this aggregation mechanism also can be analyzed by using the coarse-grained model. And then you have some kind of a mesoscale, molecular dynamics. I didn't really use it, but you are just approximating the protein, kind of a surface of the protein is kind of a mesoscale structure of the protein, but it's not really very widely used in the protein ligand interaction because the proteins are very simplistic in terms of the structure. So it's really hard to understand what is really going on. Almost impossible at the molecular level. Therefore, they are not really suitable to be implemented in a protein ligand interaction. So these are the molecular dynamics limits. There are some other limits also in protocols, but I'm not going into details right now. The time limits are quite nicely solved, at least in the analysis of protein ligand interaction. So we could reach, even with the latest GPU, the microsecond time scale, which was not feasible 10 years ago. And for instance, for 10 years ago, the standard for the protein ligand interaction was only like 50 nanoseconds to be produced by using molecular dynamics for at least for 2 or 3 weeks. So today we can reach this level in 1 day. So a little bit of history. Molecular dynamics was the method of the molecular of this history of this method was originally developed in early 50s together with Monte Carlo simulations. The methods were developed in the US and in the laboratory in Rosenbluth and Metropolis. So they started to implement this method to analyze time evolution of n-body systems using Newton's equations of motion. So some algorithms were analyzed to investigate the celestial mechanics, focus on the celestial mechanics and stability of the solar system. So this was implemented before the molecular dynamics was used in the molecular research. Then, when the supercomputers were emerged, the molecular dynamics became the method of choice to be implemented for theoretical physics, for material science, biochemistry, to be frequently used to refine three-dimensional structures of protein and different macromolecules. After from the X-ray crystallography and NMR spectroscopy. So, actually, we are also doing similar things here. We have the protein database, data bank, then we just download the protein of interest and try to simulate this protein in a water environment. Usually, the molecular dynamics is a kind of a post-processing method when you have already proteins with ligands, so you have some preliminary results from the protein docking, and you will try to confirm the results by using molecular dynamics. If sometimes the results are different, then you have to redesign the pipeline you have. And we are also implementing the same strategy here for our proteins, for the GABA protein as a receptor and for ghrelin receptor, where we already have some results from the molecular docking. So implementing the molecular dynamics would be the confirmation of our previous results. Sometimes molecular dynamics is also widely used to simulate proteins and ligands. It can be also used to simulate some thin film growth, ion supplementation in nanotechnology devices. You could also simulate the different surfaces in tribological experiments. You could also simulate ab initio prediction of protein structure by simulating folding the protein folding of polypeptide chain from the random coil. And we also did these experiments when we were trying to analyze the folding of the trip cage protein. This is the relatively small protein, only about 20 amino acids, and it has one alpha helix and one beta sheet with the coil with the tone which connects these two structures. And this protein can be very easily simulated in the molecular dynamics. The results of the molecular dynamics can be compared with the predictions and with other predictions, and can be tested through community-wide experiments in the critical assessment of protein structure prediction, some kind of CASP experiment. The critical assessment of the protein structure prediction. So we could have a break and then we will start in 15 minutes, okay?
Okay.
High performance computing is required. So if you would like to do the molecular docking experiments, it's easily performed on the standard laptop computer. But for the molecular dynamics, you need the high-performance computing facility, usually the cluster, and sometimes the supercomputer with multiple GPU, CPU units, and some graphic accelerating programs like CUDA modules are also required. Therefore, one cannot really perform this analysis without the proper settings. And the simulation should be performed at least for 1 week or so. So it would also requires a kind of a maintenance facility like we have in Germany. We have a cluster to run the simulations, and the cluster is also maintained in a specific room, in specific cold room, when you can cool down the CPU and GPU units. This is also important if you would like to run some algorithms which are based on the hybrid techniques like quantum mechanics, molecular dynamics simulations, where you would like to combine these two techniques. One is the all atom molecular dynamics, and the other is quantum mechanics. The only thing is you have to combine this, like you have the ligand and the binding site. Usually this binding site represents by some small number, relatively small number of residues. Then this area can be simulated by using the quantum mechanics like the DFT, and the rest is simulated by using the all-atom molecular dynamics. These techniques are also quite time-consuming, but they are possible. In our facility, we have all these programs to perform this kind of hybrid simulations. So the molecular dynamics is also very promising methodology, and couple of years ago, to be precise, in 1999, it was the main topic to discuss, and the Nobel Prize in Chemistry was gifted to the scientist who developed the first formulations, first algorithm for the molecular dynamics, such as Michael Levitt, who shared the Nobel Prize for the application of molecular dynamics. And he wrote that the central embarrassment of molecular mechanics, namely that the energy minimization or the molecular dynamics generally leads to a mode that is less likely the experimental structure. So right now, the molecular dynamics is considered as a molecular microscope through this kind of computational in silica molecular microscope. Through this, one could analyze and simulate different molecular phenomena, such as protein-ligand interaction, protein-protein interaction, and some other mechanisms like protein motion, aggregation, folding, unfolding, among other things. Molecular dynamics is also important to be integrated in a different chemoinformatics pipelines, like pharmacophore development and drug design. And people are trying to combine these two to see how, for instance, we already discussed previously what the pharmacophore is and why it's important to analyze different pharmacophores. Here we have some kind of a technique to compare different pharmacophores during some period of time, and probably this would enhance our understanding on the protein-ligand interaction. So, as I already mentioned the molecular dynamics is a force field technique, which means that all the interactions, molecular interactions in the system, they are programmed in a kind of a force field protocol, and for the force field, different force fields. So you have various force fields for different molecules, and here just an introduction of a general idea what is the force field, what the parameters represent the force field. So the force field is actually all the interactions which are provided by the pairwise interaction, chemical bond interaction, and different potentials. So like we have bonds, we have angles, we have proper dihedrals and improper dihedrals, and we have different potentials like van der Waals electrostatic Coulomb constant. This is for electrostatic potential, and this is for van der Waals potential. So these are the most important parameters that we have in the force field. So the force field actually is a file where you are keeping all the information about the chemical bond interactions and the pairwise interactions. So this is the most important information that you would have in the force field. And then you get different solvers also, which contain the radium quantum number for electron state, some constants. So they are used in the force field with charge depots and charges and depots and etc. So what kind of force fields we have? We have different force field. The type of the force fields can be for the protein, like FF90SB. Sometimes we are using proteins sometimes we're using force fields not for the proteins but also for the DNA, like OL21 for RNA, OL3 for carbohydrates. For carbohydrates, we are using glycam force fields such as glycam 06J, and this force field is the main force fields, for instance, how to simulate the interaction of cyclodextrins. There are also some force fields for lipids, like lipid 21, and for some organic ions as well. You also have water models, and water models include TIP3P, SPCE, TIP4PEW, and OPC models, which are integrated in this force field, so you can actually combine this information and use it together with different force fields. These are the newest force fields. Of course, you have different outdated force fields, like FF99SP. It's so far the most tested force field implemented in molecular dynamics protocols, but it's a bit outdated. So now we have FF19SB, which is kind of promising in terms of the improved pairwise and chemical bond interaction. Force fields are sometimes creating some kind of artifacts. Therefore, to use the force fields, one should have a very good parameterization with different molecules to produce the results, which are very close to the experimental results. Sometimes these force field parameters are matched to some functions like a radial distribution function to be analyzed, and at this point one could say that this force field is suitable for the simulation. Protein force field is the most widely used force fields like FF90SB, and it has the improved amino acid dependent property such as helical propensities and reproduces the differences in amino acid specific Ramachandran maps using amino acid specific SMAPs parameters. So actually what you have here, you have the Ramachandran maps taken from the experimental results, and by implementing the software you could actually reproduce these results which are very close to the experimental data. This force field also has a very specific backbone parameters. So far, it can be used with OPC water model. If you want to try the oldest version of this force field, such as FF99 force field, then you have to use TIP3P model, water model. And this water model is again the most extensively used water model for the molecular dynamic simulation. You have different modifications of this model, like FF14SB, FF1 LISC-SD, when you have, for instance, improved parameters using quantum mechanical data to fit quantum data. Some sidechain quantum dihedral parameters are improved where they fit better to the quantum mechanical data for each amino acid. These force fields are widely used in an EMBA molecular dynamics pipeline. We have different molecular dynamics software. For instance, GROMACS. In a GROMACS, you have different force fields. So this is the representation of the different force fields and how one could implement them, just to give you a short overview on the ability of these force fields to be integrated in the molecular dynamics. So as you can see here, we have different sorts of the coarse grain force fields, which are representing the molecular part of the molecular part of the virus as a protein, as a single protein, and this protein was represented by the coarse grain structure here. And you could actually simulate the whole capsid with the RNA inside. The other simulation is the virus with some spike proteins. Here, as you can see, it has a very big shape, like 75 nanometers. And again, the protein structures are represented by the coarse grain here, like this one. And after after compiling all this, after generating all the capsid structure, you could actually simulate the dynamics of this virus. You could also create some coarse grain for the lipid membrane, where you can approximate choline, phosphate, glycerol, ester, and tails with coarse grain structures here, coarse grain sites. You have in total you have coarse grain sites, and then we will analyze it using the finite element landscape. Then we will see that the coarse grain structures are also in agreement with the finite element representation for the all atom structure. At least you have here the global minimum where you could reach the minimum energy, which is the same as on the all atom finite element plot. There are some other techniques to be implemented in molecular dynamics. One of them is the technique to simulate the protein ligand interaction free energy, and it's called the linear interaction energy. When you have some sort of kind of a circle, how you can calculate the Gibbs free energy of binding, and this Gibbs free energy of binding can be determined by calculating the energy for each of the parameters representing the system. Like you would calculate first for the ligand, then you would calculate for the complex, then you will calculate for the protein, and after that you would implement the equation to calculate the protein ligand interaction energy as the Gibbs free energy of binding here, as written here. So you get some differences in the Gibbs free energy. This analysis is widely used when you get delta G. Bind is widely used when you have the estimation of the impact for some mutated amino acids on the protein structure. Then you would have a different energy for the wild type residues in comparison to the mutated residues. Then the difference between these two entities would represent delta G, and it would give you the information whether the mutation would be detrimental for the particular protein structure. There are also some other techniques which can be implemented to calculate the energy of binding, like MMPBSA, GBSA, and the energy perturbation technique as well. You could also implement the hybrid techniques to use the quantum mechanics to calculate the Gibbs free energy of binding for this particular system. So what are the molecular dynamic software, and what are their benefits and drawbacks, and how we could actually choose the right software for our simulation? These are just a small number of the options we have, but this software is widely used to simulate the molecules. The first is the open source. This is the GROMACS molecular dynamics software, the Groningen machine for molecular simulation, which was designed in the Netherlands, in the University of Groningen in 1991. The software is free of charge. It's the open source, so one could use it. And now it's also running on the CPU and the GPU modules, and it is primarily designed to simulate different proteins. So it's not really very well adapted to the protein ligand interaction. You have to use different external modules to generate the topology for them, and then trying to integrate into the EMBA, into the GROMACS software. So it's a little bit tricky to do. There is also not very good protocol to generate protein ligand interaction. Most of the organic molecules are not very well suitable in GROMACS, but the GROMACS software is probably the best if one could simulate the protein complexes or protein dynamics at different time points. So if you have the dimer, or if you have the protein interaction project, then the GROMACS is the best software to simulate these dynamics. They also have a lot of utility tools. It's very easy to implement the analysis in GROMACS, very straightforward. They have also some very advanced parameters techniques like umbrella sampling and some alchemical calculation protocols. You could also add some plugins like Plummet plugin for the free energy calculation. So the program is quite scalable and has a lot of analysis tools. The other software is the EMBA. The EMBA is the assisted model building with energy refinement. Was initially designed at the University of California in 2002, and by my experience, is the best so far. It's the best software to investigate the protein ligand molecular dynamics, and you could run it on the CPU or on the GPU. It also has the CUDA integration, producing the module which is called PME, Spatial Particle Mesh Molecular Dynamics. CUDA, the module is quite fast. So what they did, they took the sender module, removed all the unnecessary and time-consuming protocols, and added the CUDA module to this to run on the high-performance GPU graphic cards. And so far, the speed is probably the best, but the best you can get for the protein ligand interaction in comparison to similar molecular dynamics software such as GROMACS. So the EMBA is also very scalable. You could add different modules. Also, you could add Plummet, and it has also the scripts to calculate MMPBSA, GBSA gives free energy, and you could generate the protein ligand interaction, and if it's the receptor, membrane receptor, then you could submerge this receptor into the lipid membrane. There is also a software, the part of the EMBER pipeline, which is called EMBER Tools. Presently, you have EMBER Tools 23 and EMBER 22, which is free of charge. So it's good news for the academia, because in the past you have to pay like $500 for the license. So today it's completely free, and you could use this software without any limitations. The software has all the force fields I already mentioned, and it also has compatibility with some other external tools to generate different complexes, like charm GUI, or PACMO memgen. Actually, this PACMO memgen is also a part of the EMBER pipeline. On the other hand, you could also implement the Maestro, the Schrödinger Maestro protocols to create the system, and after that, you could simulate it using some converting tools. You also have very elegant trajectory analysis tool, which is called CPP-Traj, where you can calculate different parameters like RMSD analysis and RMSF, RMSD analysis, the number of the hydrogen bonds, and radius of gyration, and some other parameters like displacement analysis, and so on. You could also calculate the PCR, the principal component analysis, and Markov state model. So it's actually pretty powerful. So you could calculate a lot of things. It also has the free energy workflow tool, which is called VEW, and it has also the non-equilibrium free energy toolkit for the EMD module. Quite powerful. You could also simulate not just the proteins, but also the ligands. Sometimes you could even you can create the topology for the different polymers. We could also simulate some kind of extracellular matrices, like a bone matrix, which is called hydroxyapatite. So this was a project when we simulated the hydroxyapatite surface, and using this particular methodology from the EMBER pipeline, and after that we use this as a receptor for molecular docking studies to understand the interaction of protein ligand hydroxyapatite interaction using some antibacterial substances. So it's actually pretty good, and this software was largely developed by Professor Coleman, starting from 1975 to 2001 at the University of California, who was the main contributor to this program. And right now there are a lot of developers and also contributors who maintain the open source code and could share it to the different universities all over the world. We also have it, and this software is still under development. And actually, this software we are pretty happy with. And the other software is called NAMD software. And then this software also widely used software for molecular dynamic simulation, for the scalable molecular dynamic simulations. And this software is the abbreviation is the Nanoscale Molecular Dynamics Program, which was developed in the US, University of Illinois in 1995. Also quite powerful software. We could use it in our studies. This software was also implemented for various nanobiotechnology projects, and we have some projects using this software. We are trying to implement some kind of next generation sequencing, where we're constructing the pore, the nanopore, and through this nanopore you can try to squeeze the DNA molecule or the RNA molecule to see what kind of signal you would get, like the one you have in a nanopore next generation sequencing platform. This NAMD and AMD software as a scalable molecular dynamics is also used to simulate various systems, various biomolecular systems. You have also the interface, which is called VMD, Visual Molecular Dynamics Interface. You will just download the software to this Visual Dynamics interface, and you would start to analyze, process the protein, and you could generate the structure to be processed using the NAMD. Right now you have a CPU, GPU compatible NAMD modules, and you could also, sometimes files are compatible with Ember, Charm, Explore, so also quite compatible. You could actually convert some files. You could, if you have some trajectory from GROMACS, it's also very compatible, at least using VMD to analyze the GROMACS data. Sometimes I am also using this VMD to analyze various molecular dynamics data, such as GROMACS. The binary is free of charge, so you could also use it for free. And you could find a lot of tutorials like nanobiotechnology, which you can find on the Alek Aksymentiev's website, where they put in a lot of tutorials concerning this nanopore. They also have the simulation of protein ligand, protein submerged into into the membrane, but this protein is very complex. They simulate the transporter, the bacterial transporter, and how this transporter actually is transporting the ions inside and outside of the bacteria. So also a very good software. The other is the Desmond software. Desmond software is also scalable at multiple GPU, CPU, and was developed by the Shaw Research in the US, Research Center in the US, to perform high-speed molecular dynamics simulations by using the conventional computer cluster. They designed the code using the parallel algorithms and numerical methods. So I think we can have a break and we will continue with the software later on. Okay. Very efficient, and it's now integrated as a commercial software through the Schrödinger program. The Schrödinger program has this software installed as a part of the Maestro module, and is using the particle mesh event-based methods as well, similar to previously described molecular dynamics methods. Therefore, it's compatible with the Maestro. You could create the system in the Maestro interface and then transfer it to Desmond. It also runs on the GPU. We also have this software installed, so those of you who is interested in long-range molecular dynamics could use this software, and it accelerates. Its accelerated version is about 60-80 times faster than the central processing unit version. So quite remarkable speed one could achieve using the molecular dynamics. Desmond also quite efficient for free energy calculation, such as free energy perturbation, FEP. It has also a FEP module in the Schrödinger pipeline. The other simulation methods, such as replica exchange, are also supported by this algorithm. And some force fields also compatible with this software, such as CHARM, AMBA, OP, LS software, and range of different water models. And this software was designed to be used by Shaw Research, by the Shaw Research Center on the cluster, which is called Anton. And now there is a second version of Anton available, which has the latest GPU units. And the last software I would like to talk is CHARM. It's not really very widely used right now, and as a kind of for the historical reasons, one could mention this software. Not really very powerful, not up to date, because doesn't really, as far as I know, it doesn't really have a GPU integration. So, but in 90s, in the end of the beginning of the beginning of 2000, it was used to simulate relatively small system, but now it's obsolete, so it's just for the historical reasons. It's kept for the historical reasons, yeah. And as an example, I would like to show you also some information, some facts on the molecular dynamic simulation about the interaction of potentials and forces, interactions between atoms and molecules, results from electronic structure. Not a classical problem, requires quantum physics. Therefore, two different ways to proceed leading to two different classes of molecular dynamics can be devised. The first one is the classical molecular dynamics, and the other is ab initio molecular dynamics, when you could determine all the information from the quantum mechanics equation. There is a software which is called TerraChem software, which is using von Oppenheimer ab initio molecular dynamics on the GPU. One could also try to implement this for some very small, for some small proteins, like the charge transfer within the protein. This week there was a publication this week concerning the charge transfer in the proteins, published in Science. So one could also use ab initio molecular dynamics to create a model of this event. The classical molecular dynamics uses interactions which are approximated by classical model potentials constructed by comparison with the experiments. They are called the empirical potentials, and it leads to the simulation of purely classical many-particle problem. The classical molecular dynamics works well for the simple particles such as gases that interacts via isotropic wave potentials. And poorly covalent atoms and metals can be used as well, such as electron form Fermi gas. Simulations are usually fast, permitting the large particle number up to hundreds of thousands of atoms. In comparison to ab initio molecular dynamics, which is performed as a full quantum calculation of the electronic structure at very time step for every configuration of the atomic nuclei. The forces are found the independence of the energy and the particle position, and much higher accuracy. It has a much higher accuracy than classical molecular dynamics, but much higher numerical effort, which restricts number of particles and the simulation time. So last time I already mentioned the simulation time of the ab initio molecular dynamics is only in the range of picosecond, femtosecond, picosecond range. And the atomic number is restricted to some hundreds, at best thousands of atoms. So here, as you can see, we have some CPU and GPU supercomputers and clusters. We have this Christofari cluster supercomputer in Russia. we don't really have the access to the supercomputer. So for probably it was built for some commercial reasons. Therefore, it's just it's kind of a publicity stunt because we still we didn't really have any experience with this supercomputer. There is also one supercomputer in Germany. It's called Julia. It's the GPU supercomputer, and I have the access to the supercomputer, but the access is limited because it's always fully occupied, and the time is limited to run the program. This is just an example of the Anton supercomputer developed by the Shoah Research Center to perform high-speed molecular dynamic simulation using the Gizmo software. And this is the cluster. This cluster is an exact cluster. Right now it's a bit outdated because it uses the outdated CPU graphic cards, and right now we have the 4080 GTX GPU graphic card, which excels the old graphic cards represented on this cluster. So this cluster is also fully functional, but in terms of the speed, it's more than 10 times less efficient than the new high-performance computer we have right now. So what kind of projects we have, and what are the methods we are using for the, such as molecular dynamics or molecular docking. I just want to point out one of the projects where we were trying to design the different biologics using molecular dynamic simulations. So these biologics are peptides. It's called a T-peptide as the HIV inhibitor, and this T-peptide was discovered in 1986 at the NIH, and this is the synthetic peptide. This peptide T-derived from dapta peptide is a drug. Is a drug in the clinical trials which blocks the binding of the CCR5 receptor. The HIV binds to this receptor to the CD4 receptor by means of the CCR5, and when you get such as on this picture, and if you are going to block the CCR5 receptor, then you will interfere with the process of the HIV anchoring to the plasma membrane, so the virus wouldn't have a chance to go into the cell. And this mechanism could be reconstituted by using the molecular dynamic simulation, using the T-peptide and some receptors like CCR5, or some other compounds like cyclodextrin, if you would like to increase the solubility of this peptide. So this peptide T showed several positive effects related to the HIV and the neuro-AIDS. This peptide was associated with improved performance in the patients suffering from the severe cognitive impairments. The HIV has different very complex structure. It has also a capsid and some proteins which are interacting with the CD4 peptides such as GP120 and GP41. So these are the main peptides which would interact with this receptor, and the co-receptor helps to get the perfect alignment of the CD4 with the GP120 variable loops. And the project was related to the pharmacokinetics of the T-peptide, where we were trying to formulate it to improve the stability, its stability, and solubility. And for this, we were using the beta-cyclodextrin. The beta-cyclodextrins are oligosaccharides. They used to improve property, stability, solubility, drug release, and so on. The beta-cyclodextrins are widely used in the formulation of various substances. They have the 7 glycosidic units with the mildly lipophilic binding cavity and hydrophilic exterior. And this exterior is mostly organized by the hydroxyl group, and the interior was formed by the glycosidic units. So the idea was to perform the formulation using the molecular docking, and then in the dynamic simulation, one could analyze this dynamics and how the T-peptide would interact with the cyclodextrin structure. So the cyclodextrin structure has a very complex topology. It has a primary phase and a secondary phase, and the primary phase has a higher number of hydroxyl group, up to 14 hydroxyl group, and the secondary phase has the smaller number, only 7 hydroxyl groups. So these hydroxyl groups can be substituted. The T-peptide, which is found here, has some lipophilic residue like tyrosine with the lipophilic residues, and most likely this residue is the one who interacts with the mildly lipophilic cavity of the cyclodextrin. The cyclodextrin is also stabilized by the intramolecular hydrogen bonds within the T-peptide structure. So how we could actually analyze this T-peptide? We could analyze the deviation of this peptide in the solution to find some highly flexible and less flexible parts of these peptides. So when we will put this peptide in the solution, it will utilize some kind of saddle-like conformation where the C-terminal and N-terminal and C-terminal parts of these peptides are highly flexible, and we could perform the root mean square deviation to measure the molecular dynamics, stability, and flexibility of these peptides by least square fitting of the peptide to the reference. So this is the equation where m is the mass of the peptides, and the mi is the mass of the atom, and rt is the coordinate of the atom I of conformation at a particular time, and rit is the coordinate of atom I and at the initial state t0 native conformation. So actually we calculating the root square deviation by using the native conformation as a initial structure here at the initial state 0. Here we also there are some equations how we can determine the equilibration and dissociation and association constants. So I would just give some overview on what will happen in a protein with a ligand cyclodextrin system. And in this system, you get the binding when you have when it's described by the binding, the kinetic constant, binding constant, or you have a dissociation if you have the Ki or dissociation constant. So this is the equation. They usually reverse the binding constant is the reverse to the dissociation constant and vice versa, and you could calculate it as a logarithm of binding negative logarithm of a dissociation constant. The binding energy is calculated by using the standard equation, either using the binding constant or using the inhibition constant. From this equation, you could also have devise the enthalpy compensation as a difference between enthalpy and entropy. And this is the result from the molecular dynamic simulations. We used the standard algorithm. So in this algorithm, we performed either the cyclodextrin simulation or the peptide. As you can see, you get slightly higher deviation of the peptide alone, but if you would do the complexation, you would have a decrease in the deviation of the peptide. So here you can see at least at 60 nanoseconds we have before 60 nanoseconds you have a significant decrease, and then you have the increase. Then you would have to run further, at least for probably for 500 nanoseconds to see what's going to happen next. Okay, so this is the complex stability, which is correlated to the number of hydrogen bonds in a peptide and in the complex. You can see that the peptide has propensity to form more hydrogen bonds with the solvent, which is not the case for the complex, where the peptide is protected by the cyclodextrin molecule here. And this is the energy evaluation of the cyclodextrin peptide complex from the electrostatic interaction and the Weiss interaction as a Leonard-Jones potential. You could actually calculate as a sum, you could calculate the interaction energy, and the interaction energy is presented here. You could also, if you would have different system, you could also compare different interaction energy to have some idea what is the ligand which performs best in terms of the binding. Well, we could also calculate the buried area, and this buried area was calculated by using this equation, and we calculated using the area between the cyclodextrin and peptide T to understand, to describe the interaction interface. Here's the representation of the buried surface area using the 2D plotting, some kind of a lit plot software, and you could use the external program which is called an access to calculate and plot the buried surface area. And thank you for your attention. If you don't have any questions, I will see you at the next lecture.
And